Dr. Ray here. I'm here to give you a short video on development of the spinal cord today. We already went over the development of the neural tube in the last video. If you haven't watched that one, you might want to watch that one first. And so you understand that there was a neural tube that developed from induction by the notochord. And then this neural tube will close at the cranial-caudal end eventually. And the cavity inside the neural tube is important because it will develop into the ventricular system of the brain. So in the neural tube region that does develop into the brain, the cranial aspect of the tube, the cavity inside will become the ventricular system, and you'll be learning that when you learn brain anatomy. The central canal, the spinal cord, is very small, so it's going to remain a small cavity after full development. So at the beginning, this neural tube looks like this here. You can see this embryo, neural tube's already developing, but the embryo is still kind of flat. It's coming, starting to fold, and you can see the separation between the lateral mesoderm, the splanchnic and somatic or parietal and visceral layers here. And this is the region where the neural tube is developing. And in the adult, this is what the neural tube in the region of the spinal cord is going to look like. And so at the beginning it's rather homogeneous. There's a bunch of cells. It doesn't really have a lot of organization. But then this neural epithelium of the neural tube will further segment out and specialize. And as it does that, it reduces the inner canal from being a wide open canal to a very small central canal of the spinal cord. So in this picture, you can see a closer up view of the neural epithelium, and you can see that these cells are really packed in here, and these are going to be the cells that will develop into the neurons of the spinal cord and also the macroglia. And so this is from the textbook, and they show you that these neuroepithelial cells will start dividing, and at the beginning there really is just a thick layer of these neuroepithelial cells. But then what happens is that those cells migrate into other zones, outer layers, and here's the neuroepithelium inside. They call this the ventricular layer because it will align the central canal and ventricles at the end when these cells are finished with their proliferation. But essentially these cells are going to give rise to all the neurons that will stay in here in this more intermediate layer, and they will grow a bunch of axons that will extend into what they call the marginal zone. Now there's not only neurons and axons, however, there will be proliferation after the neurons have been generated from the neuroepithelium. The neuroepithelium will give rise to the glioblast, so the macroglia that exists in the marginal intermediate zones all come also from this neuroepithelium. And then I want to point out that what we mean when we say macroglia is that astrocytes and oligodendrocytes. So we haven't really gotten to the central nervous system, but we sort of go with systems. So since you're learning about this development, you can already start to be familiar with there are different types of glia. The macroglia are astrocytes and oligodendrocytes. Microglia actually do not come from the neural tube, they come from bone marrow, and so that's sort of an important part to hold on to. All right, and so this proliferation of these neuroblasts are going to aggregate in certain areas, and it's going to cause this neural tube to sort of reshape itself and have sort of a thin roof and floor and thicker sides. That's how we get the spinal cord looking the way that it does in the adult. And what another structure that actually becomes evident is a little sort of divot in the spinal cord that we call the sulcus limitans. And this groove is going to be the place that separates the functional changes that occur in the neurons that exist in the more dorsal side and the more ventral side of the spinal cord. And so the area that's on the more dorsal side of this developing spinal cord, we are going to call that aggregation of neurons the alar plate, and that those neurons have afferent functions because we have connections with the spinal ganglia, or dorsal root ganglia, coming into that region. And then the other plate we're going to call the basal plate, and that has efferent functions because these neurons actually are sending signals out to move skeletal muscles. And so the alar plate are where the cell bodies are located have lots of functions, but one of the functions is to receive information from the dorsal root ganglia that we talked about in the neurulation video that came from neural crest cells. And so they're going to eventually become the neurons that are in the dorsal horn. Okay, so here you can see the signals are going to come into the dorsal horn, and so when you're thinking about the spinal cord, hopefully forever, you will think about how this is functionally different on this dorsal side to the ventral side, and you'll learn more complexity in your neuro course. And then don't forget that the dorsal root ganglia, also referred to as spinal ganglia, all came from neural crest cells. Remember, their signals come in, and the cells that they're actually going to be synapsing with are the cells that came from the alar plate. Therefore, they came from neuroepithelium. Okay, the basal plate, these cells are going to have efferent function because these neurons are going to send signals to skeletal muscle, and so it's going to become the ventral horn and the lateral horn of the spinal cord. Okay, and so when you look at the picture, it's going to be located in this region. And these signals go out. The axons grow out along with the musculature that they are their effector targets. And so another important thing that you need to know about the spinal cord just as it relates to development is at the beginning of development here, this neural tube is just as long as the vertebral column. And so all of the nerves that grow out are going to exit at the same level as the vertebra. But then over time, the vertebral column actually grows faster and longer than the spinal cord doesn't grow anymore. And so although you have connections of the conus medullaris all the way down to the coccyx with those terminal phylum that you looked at in lab, the nerves are not going to end up exiting at the same vertebral level because there is a stretching of the roots as the vertebral column grows. And so that's how you see the end of the spinal cord way higher up than the end of the vertebral column. And this is all because of development. You can see the different phylum terminali. There's the internal one inside the dural sac, and then the external one goes all the way down to the coccyx, and that is going to kind of hold the spinal cord in place, and that was the original length of the spinal cord. And so at the end of development in an adult, the spinal cord ends up becoming approximately at the level of the intervertebral disc between L1 and L2. However, hopefully you are beginning to appreciate through dissection that there's anatomic variation. So it can be as high up as T12, as far down as L3. And all of the roots that extend down because the vertebral column grew are going to result in that cauda equina. Okay, that's the most important things that you need to know about development of the spinal cord.